All right. Well, thanks very much, Dan, and everyone for having me here. And very excited to talk about my PhD work. My thesis will be put on the MetaUni Discord server on one of the chats there. I'll let you know. I think we'll let you know where that is later. I'm a PhD student at OIST in Okinawa, Japan. OIST stands for the Okinawa Institute of Science and Technology. It's a relatively young university. It only has PhD students, and it also has interns. If you're interested in spending a little bit of time in East Asia, definitely recommend coming and doing an internship, which are fully funded, by the way. So you've got a lot of opportunities to, it doesn't matter so much about your financial situation. It just matters about your passion and your science. So check it out. My thesis is broken up into 4 chapters, and over the following 4 weeks, I'm going to talk about each of those chapters in order. And as we were talking just before starting today, I was explaining that actually the sort of the narrative of my thesis is not entirely linear. In fact, I actually put in the introduction, the end of my introduction, that thesis can be read in any order. So even though that I'm giving these talks in the order that they are, each one is sort of self-contained. And running through all these talks is some general theme of geometry and topology and neuroscience and AI, and in particular memory and navigation. So I hope that this is interesting, and let's begin with the first talk today. So this talk is includes a paper that's available on eNeuro, which is also just in the thesis. So you can have a look on Discord for that. And it's titled A Functional Role in Inhibitory Diversity and Associative Memory. So firstly, let me direct you to the second slide, which is just, I don't expect you to look at the detailed picture, but it's just sort of a nor for the third slide when it comes to, I expect you to look at the detailed pictures, but it's really just to illustrate that there is a lot of diversity in inhibition. So so neurons in the brain actually have at least 2 different types, broadly speaking. One are excitatory and another inhibitory. So excitatory neurons send out positive signals, basically, to the neurons they connect to, whereas inhibitory neurons send out inhibitory neurons. We call this distinction Dale's law, and it's actually being broken slightly in some cases. So there are some neurons where they actually send out a mixture of inhibitory and excitatory signals, but it's very rare. In general, this principle or law, Dale's principle, Dale's law, is held throughout the brain. What's interesting is the excitatory neurons are relatively simple or similar across their different instantiations, whereas the inhibitory neurons are quite wild and varied. There's a bit of a zoo. So, for example, in barrel cortex, which is a part of the brain in rodents which takes the main input from their whiskers, like their whisker sensations, that area has at least 14 types of inhibitory neurons that we can tell, like based on their shape and their genetics and things like that. And in visual cortex, there are at least 28. So moving on to the next one, next set of slides, I'll just walk over here. So, and another reason why inhibition is interesting is because it's also quite consequential for a whole bunch of diseases and problems that people experience, such as autism, depression, Parkinson's disease, schizophrenia, and the list goes on. And the main reason for this is that inhibition very simply sort of controls and limits excitation. So it's kind of a yin-yang situation for the brain. And if you have too much excitation, you get things like epilepsy, like it's uncontrolled. And if you get too much inhibition, you might get something like depression. At least that's one theory of depression. And obviously there are many different types and etymologies or causes depression and other diseases. I'm not saying inhibition is the sole and only cause of all these diseases, but they're heavily implicated. So, given that inhibition is so diverse and so consequential, let me just give one concrete example based on some of my previous research. So, this is a paper that's available in a journal called Physiological Reports, and it's about traumatic brain injury. And what we did is, this was done at Monash University in Melbourne, and what we did is we took rodents. This is on the second slide, or the fourth slide that is on the right side. Let me just check the orb cam. Yes, that can be seen. So what we did is we took rodents and we actually gave them a diffuse traumatic brain injury, which is a little bit cruel, but they're under anesthesia when we do that. And then what we did was we also put them under anesthesia and measured the neurons in barrel cortex. That's the area which is responsible for picking up signals in the brain for their whisker sensations. And then we touched the whiskers in a systematic way, according to on that slide you can see in the top left. And I'm going to experiment with this tool if I can. Oh yes, highlighting this top thing, just putting a green box around it. That is a waveform of the deflection pattern that we put onto the whisker. So if I just highlight that in red, this is like going down and then up and then sort of wiggling around. So the whisker is being deflected in that sort of pattern of motion, in that one-dimensional motion, starting from a resting state. And then we can change the amplitude of that waveform. So that's what's happening on this y-axis. And then on the x-axis is time. So, and then here you can see different sort of colors. The higher area of this color bar means a larger firing rate of a particular neuron. So this is just for one neuron. And then blue is just no activity. So neurons have activities, they spike, they go on and off, and you can sort of calculate the rate in which they're going on and off, or the rate in which they're sending signals. So you can see as we increase the amplitude in this case, you get differences in the responses, and we were categorizing these responses as sort of like profiles overall. And in this particular profile, you can see there are two fairly strong components happening at about 10 milliseconds and 25 milliseconds into the stimulus, and that actually, you could line that up to different parts of the waveform there. Now what we saw was, given prior evidence that traumatic brain injury dramatically perturbs inhibitory neurons, preferentially, especially diffuse traumatic brain injury, which basically just means kind of a rush of fluid forward and then backward in the head. It's kind of like what happens in a whiplash injury in a car crash.
What's the reason for that?
In that
damaging
case,
inhibitory neurons specifically?
Sorry, could you say that again?
Why should that affect inhibitory neurons more than excitatory?
Typically, so I mean, we don't know exactly why, but we guess it's mostly because they're smaller and their structures are finer and more delicate. So when the fluid rushes forward and backwards, it's more likely to damage the cell membrane.
I see.
And then it sort of tears them apart.
What's the relative proportion? Is it like the overwhelming majority?
I think it'd be something like 80% of cell deaths are inhibitory.
I see. Interesting.
Yeah. It might even be more than that. Yeah. And so we were interested in the functional implications of that for the excitatory neurons, since these responses are being recorded. We think, we don't completely know, but we assume and it's quite likely that they're from excitatory neurons. And so then we're looking at the response profiles and their sort of components over time and over disease state. And I won't go into all the details, but in general, you see a progression over time over a period of up to 8 or 12 weeks, which we looked out at, where this the timings and number of components in these responses change quite wildly compared to the healthy controls. And so we posit that this is mostly due to the functional role of inhibition, and you would otherwise not necessarily be looking at this sort of level of granularity. You're normally when you're looking at inhibition and excitation studies, you're looking at some general balance of things, some yin and yang, but here we were looking quite detailed into the sort of individual neuron responses, and we found that there were quite dramatic differences, which eventually sort of had a level of functional recovery, it should be said, for those animals, for some of the animals, but not completely. So should there be no further questions, I'll move on to the next set of slides where I'll start talking about memory. Okay, yeah. Oops, I'll just move here so the orb cam. Yeah, good. All right, so now let's talk a little bit about memory. So hopefully now I've established to you that inhibitory neurons are quite important. Let's just talk about excitatory neurons. So this slide on the left shows a few things. On the top left, we have an area of hippocampus. So this section here is hippocampus, and hippocampus in the brain is typically where we think most of navigation, not most of memory, but it's very involved in memory. Yeah, I should say long-term memories in the rest of the cortex, we think, but it's highly involved in basically processing memory and navigation.
Could you just say like two sentences on the sort of basis for why we know that? Like if your hippocampus is damaged, you can't navigate, or what's the...
Yeah. So I think some of the main evidence comes from human studies where they remove part of where they've had like a lobotomy, an accidental one or intentional one for epileptic reasons, where they've removed part of the hippocampus and they've suffered greatly in terms of their capacity for navigation or memory. There's a very famous case called HM. That's the initials of the patient, and they had a problem in their hippocampus, and they basically couldn't remember anything beyond a few seconds. But their long-term memory was intact because the rest of their cortex was intact. So, and also they weren't able to navigate around very effectively in the world. But also we have more systematic studies in animal models where we, for example, quite intentionally and specifically damage a particular area of hippocampus. So CA1 is one area, just the name of one of those areas, and then if you damage that area, it's different than if you damage some other area. It'll affect memory and navigation differently. So there's a bit of specialization, but as a whole unit, it's quite important for that. And so this is a classic study done that sort of started to look at the more details of this, other than just, okay, we've perturbed the hippocampus, but we don't know precisely what its we don't know how it's actually functioning to create memory and navigation signals. So here we have like the hippocampus, and we have these different electrodes. So there's different colored sticks, so different electrodes that are placed in different spots, and between each one of these is something like 100 micron. Oops, I'm not very good at writing with my mouse. I apologize. So and then in the picture below, this raster plot, a raster plot is just like we've got time on this axis, and then on this axis here we've got like different, you can think of them as different kind of like identities of neurons. Oh dear, neurons. Yeah, that's going to be legible. No, and so you can think of each of these little kind of lines as an instance of a particular neuron being active, and each row is a different neuron, and the colors indicate which electrode they came from. And circled, you can see that there are these clusters or groups in time of particular neurons being active all together at approximately the same time, and yet those are highly distributed over space. So remembering that those electrodes are 100 micron apart. And what this showed was that you're getting highly clustered what we call assemblies, which are distributed in space. Assemblies are neurons which are sort of becoming all active together at the same time. Another thing you'll notice is that in this picture there's something called the theta wave shown. This is just a particular low-pass filter on the electrical signal in the brain, which basically sums together all the possible electrical activity that's happening in some local space. And you can see that in this case there are troughs happening roughly at these lines indicated, which is some sort of periodic signal. And we think that, and there's a lot of evidence to support that this theta signal and others like it, these so-called local field potentials, are highly correlated with inhibitory activity. So it appears that not only do these assemblies of excitatory neurons strongly cluster together over space, over fairly large spatial distances, they're also happening in unison or concert with some sort of inhibitory activity. And we hypothesize, and there's other studies to sort of nut this out further, that different groups of these neurons, these different assemblies, correspond to different memory items. So, for example, on the picture on the right, you might have a few different memory items like a lion or a leopard or a tiger, and they might have similar groups of neurons or assemblies of neurons which become activated when presented to someone, for example, and these are groups of neurons that show up in their brain. But for the lion's neuron, it might actually share, for example, these green neurons with the tiger and these blue neurons with the leopard stimulus, and it'll also have some which are on its own. And I think, to be fair to the tiger and the lion, I think they're similar, so probably they would have some similar neurons too, but that's not shown in this picture. And so the degree of that overlap in terms of their sort of shared neurons in their assemblies is a sort of a rough corollary for their relationship in memory space. At least that's the hypothesis.
What's your intuition for how much of the information is just which neurons fire, and like, is this a very coarse indication of the content of the memory, or is it like more or less as if you knew every single neuron which fired, you would more or less know all there is to know, or you'd know like 1% of what there is to know?
In terms of memory only?
Yeah, let's say.
Yeah, I think probably you would know. I would put it on like 40% or 50% of what there is to know about memory by knowing the activity of all the neurons. And but the problem is that there's a few problems. One is that subthreshold activity of all the neurons is somewhat important. Subthreshold meaning like the activity that doesn't cause it to send an action potential or a spike. Another is that there are other cells in the brain, and another is that there's what's called drift of these activities. So if you look if you look dynamically at the system, you might notice that there's still these sort of clusters of activities representing potential assemblies of cells, but those assemblies sort of have some drift. So some particular neurons might exit that assembly in a functional sense. They might no longer become active when everyone else is active, and they might join some other assembly. And this is a problem for the stability this is like a stability issue, and it seems like actually it's useful somehow to do that, because why would the brain do that otherwise?
Reminds me of...
Yeah, this is an open...
I mean, very old computer memories used to work by continuously reading and writing. Is there some theory of memory that you sort of continuously write it back? And is that perhaps why drift happens? Or is that... Once you've written a memory, is it just stays there, or is it sort of actively maintained in some sense?
It's definitely actively maintained. It's not completely static. It's possible that the reason it's not entirely static is because that presents like a risk. Like if there's a problem in that particular area or with those particular neurons, that will have a huge effect on some important memory. But that's just one idea. It hasn't really been proven as to why that occurs.
Yeah, when Joey his talk in the festival earlier this year was also referring to drift. Just for I think maybe Chad and Ben were there, or just to recap that briefly. Yeah, so if you're trying to read out from the brain with some sort of device, what is going on and interpret it, you might run a sort of expensive learning process to figure out the correlation between brain activity as measured by your device and some sort of real world thing, whether it's a category or an image or whatever, and that's fine. So you sort of perfect that correlation, and you've learned some neural network or something that performs that mapping, but then 2 weeks later that mapping is no longer accurate. So this drift, if I understand correctly, is a big problem for getting a stable mapping from brain activity to the outside world. Is that correct?
Yeah, that definitely sounds correct, and it's made all the worse in the case of brain-computer interfaces, because you're just sampling from a small subset of the brain, so you're not able to see the whole picture. Yeah, you're not able to see where that drift is going to, and you have to pick up maybe a very small number compared to what you had before if they're drifting out of focus, so to speak.
Cool.
Yeah. So this is kind of called the assembly hypothesis of memory in neuroscience. And on the next slide, titled Temporal Association of Stimuli in Monkeys, this is a rather cool experiment done in the 80s, where a fellow by the name of Miyashita, he showed these beautiful fractal images to a monkey. So these are all the different fractal images he showed, and he was originally not interested in what I'm going to describe, the phenomena I'm going to describe, but he eventually found it later. But what he did was he showed these monkeys one of the fractals. So for example, those on the left column here, and then these ones on the third column. And then he was asking the monkeys to sort of predict something like, what's the next one? And then he was doing these in some pairs in order. But they were still being, and the way they were giving their indication about their guess was some sort of like joystick or some touching or something. And the way that he set up the experiment was actually that these were happening in quite a nice sort of regular sort of sequence. So the times between the stimuli were common, and even outside of these pairs. And what he noticed was that there were particular neurons which were lighting up for each of these different stimuli, and because they were ordered and always shown in a particular order, he could actually put these fractal images in some sort of linear sequence, and he could see that the activities, for example, of this second fractal were already starting to become active when the first neuron's activity was happening. So when the first fractal was being shown, the second fractal was already starting to sort of be expected neurally, and that's what this picture on the right shows. So on the x-axis, you've got the sort of distance away and the number of hops between the fractals, and on the y-axis, you've got some correlation coefficient, which is basically how similar is the activity when it's sort of some number of hops away compared to its maximum activity. And you can see that in general, there's I won't go through all these different lines, but these open circles are a control, so that's why they don't have this. But all the others have some sort of relationship where the correlation of activity sort of goes down to approximately 0 by about 5 patterns away. So that indicates that up to 5 patterns away, we still have some activity which is showing up in the neurons responding to some fractal, even when we're sort of 5 steps away from getting to that, and it will slowly increase over time as we get closer to it. And these were excitatory neurons, but we were curious to know, what are the inhibitory neurons doing, since we know that those are important.
Could you comment briefly on these, the ventral temporal cortex? I mean, the neurons that are firing here, are they related to what you were talking about in the previous slide with memory, or is this a different brain region that we're looking at?
Yeah, sort of, but actually this area is more to do with vision. And yet it seems that there's memory everywhere in some sense.
So if I remember correctly, the hippocampus is often involved in this kind of sequence-based stuff. Is that involved in prefetching these memories of the next fractal, or is that just baked into the memories themselves somehow?
It seems to be baked into these sensory sequences, and it sort of makes sense from a very basic neuroscience perspective, which is that the cells that sort of fire together wire together, and that has some lag. So if you have cells anywhere in the brain which fire in close succession, then the brain's going to notice that and have some strengthening of their connections. So the lag here might be, in a temporal sense, an order of 5 of those hops away in this particular area of the brain, and up until that point, there's no detection that these cells fired in close succession. So this in close time.
This one here goes backwards as well. So you're saying that if you saw B, that both A and C would be firing, or is it directional?
No, it was both. Yeah.
I see. Okay,
so it was unidirectional, and this is not showing 0, which should be like all the way up at 100 or 1.0. Yeah.
Okay. Yeah. So if it's bidirectional, then it clearly seems associative rather than predictive of a sequence. Yeah.
Right. Yeah. I would suspect. I mean, that's the report anyway. I would suspect that it is slightly directional, because that was the sequence of these stimuli, but that's a bias that I have, and it might not be supported by the data.
Yeah, I guess I hadn't understood that this idea of cells activating together, wire together. I mean, if that acquires a directional basis at some point, then it's sort of, maybe this is completely wrong, but it seems kind of reasonable to expect that as you optimize for that, you would get pretty sophisticated sequence prediction just out of that basic principle dialed up.
Yeah. However, it's unless it's done in a more sophisticated way, it's done basically in a unidirectional way. In a bidirectional way.
Yeah.
So, yeah.
Cool.
Whereas, to be useful, you would hope it's more unidirectional, since things seem to have an order.
predicting the past is less useful.
Yeah. So I'll move on to the next slides, which are, I wouldn't want to present these before the others, for example.
Nice.
And so these next slides here are just, I suppose, the one on the left is just emphasizing a bit more about inhibition. Classically, people thought that inhibition is just like a blanket of an inhibition, sort of creating some net stability of activity. And I think we've already talked about inhibition enough, so I'll just skip that.
Sorry, I may have interrupted you when you were. You talked about you did some follow-up work to the previous this temporal association paper, or you were interested in the inhibitory neurons. Is that work you did? In which case, I don't think you said anything about.
Yeah, that's what.
Is that what you're about to talk about?
That's what I'm about to talk about. Yeah.
Okay.
Yeah, so I'll move on to the next slides here, and so maybe we just skip this one about inhibition on the left, and I'll just introduce my model. So this model here on the right slide, so we're just skipping the one on the left. This is sort of a cartoon of the model structure. So each of these E circles, which I'm going to do a rough circle in red, those are excitatory assemblies representing different memories, and they can have shared neurons between them. And then these the GI is a global inhibitory group. So that's a group of inhibitory neurons which are connected to all the excitatory neurons. And this LI is a local inhibitory group, which there's also a local inhibitory group for this neighbor and for all the others separately. So there's a total of, let's say, 100 different memory items in this system. So that would be 100 different red circles and corresponding smaller blue circles attached to them, but there's only one global inhibitory group. And then what we do is we excite and we activate one of these particular assemblies. So, for example, this one that we've labeled pattern, and then we simulate for about 500 milliseconds in most cases of simulation time, and we activate this particular assembly, all of it, for about, let's say, 80 milliseconds or something like that, if I recall correctly. And then we turn that stimulation off and we let the simulation settle down and see what the activity does, which we'll see in a moment. Now these P values here, these are like the probability of connection. So the probability of the excitatory group and the local inhibitory group being connected to each other is 100% for this little module, this little memory, but then that's separated from this one. And then the neighbors of that, because we put these memories into a graphical structure, in the sense of each vertex in a graph, it represents a different memory. Then we let those excitatory neurons be 100% sort of connected to each other if they're neighboring. And for the global inhibitory group, we have a probabilistic activity or connection probability between those. And these numbers are pulled roughly from neural statistics. And across the whole network, we also add some noise to the excitatory units. And the excitatory units within a particular pattern are all-to-all connected. And then finally, we have this parameter C, which I'll show the equation for, but this parameter C basically lets us balance between how strong is the global inhibition compared to the local inhibition. So if the value of C is 0, then we're at a completely globally dominant inhibitory state, whereas if we're at 1, we're at a completely local inhibitory state. And most of the time would be in somewhere in the middle.
I shouldn't confuse that with like the amount of inhibition. It's just a different kind of inhibition, or is it?
Correct. Yeah, it's just qualitatively different in terms of the active, like where the inhibition is coming from. The total inhibition stays the same.
Okay.
Yep. All right. So let me show some of the equations for that model. So on the left side is basically a small description of the model. We start off with some static independent random patterns. So Xi is the typical symbol we use here, and I is notating the different excitatory neurons, and mu is indicating the different pattern number. So, for example, if we have 100 patterns, this will go from 1 to 100. And n superscript e is the number of excitatory neurons. And then we assign ones and zeros to this, so they sort of have a binary string. And you have the probability of one of these particular zeros or ones with f, and that f is something like how sparse or how packed is the code. And normally this f is sort of like controlling the overall firing rate in the network ultimately. Since if it's very packed, if all the memories are very dense, then this will cause a lot of activity to happen very quickly. And then, having established those random patterns, we create connection weights between neurons i's and j's. So between all i's and j's, that's all excitatory neurons to all others. And we ask, okay, for all the common patterns that you have, let's plus 1, because this will be if for example, if they're both in this particular pattern mu, then it'll be 1 times 1 here. Whereas if one of these is 0, it'll be 0. And then let's look at your neighbors. So capital K here is a graph, like this picture here. Oh, sorry, K is capital K comes from a graph, and it's the neighbors in your graph. So if we're at mu, maybe I'll do this in a different color, maybe red. So these are the vertices which are in K, and mu is the particular pattern that we're on at this second part of the equation. And I'll just do that in blue. So we look over all patterns, and then we ask, okay, what are the neighbors to this particular pattern? And then if we're neighboring, then let's also join up together. So basically, if two neurons are not only in the same pattern, but they're also in their neighbors of all those patterns.
What does it mean for patterns to be neighbors?
Sorry, could you say again?
What does it mean for two patterns to be neighbors?
Two patterns are neighbors in this graph. So so this is the memory graph, we call it. Sometimes we denote this M.
So this is just each one of these external input, this graph. I don't know where this comes from, but this is like in the brain, for some reason, or what's the reason for this particular graph, and why should I believe that patterns have a graph at all?
Well, it's a bit like the picture with the leopard and the tiger and the lion. So each one of these vertices is a different memory item, or you could think of them as different fractals, and we think that these are correlated together.
Mm-hmm.
So then we have this rule, and in the case of the fractals, it's maybe not a circle, but rather a line,
right,
because they had some sort of end. But here we just make a circle for convenience.
Okay.
Yeah. And then we have the following set of differential equations, which I don't think we have to go through in too much detail, but we can if anyone's interested.
I have a question about this general sort of framework. I first encountered this kind of thing in Hopfield's paper, but maybe it predated that paper, and this kind of statistical mechanical point of view of how to formulate these differential equations was decades old before Hopfield, or was this really, this paradigm is kind of from Hopfield, or how do I think about that?
Yeah, that's a good question. So in general, the Hopfield did sort of, let's say, popularize and come up with some of the most important original work in this area. However, there were people before him, mostly in statistical physics, like you mentioned, that were basically playing with the same equations and not relating them to neural activity as much. There were some, but they were rare.
Mm-hmm.
And so these sorts of equations are not necessarily that different from those, but in terms of interpretation and use in neuroscience, I suppose that's the novelty that Hopfield brought, and that they are based on Hopfield. So actually this first part of the equation, if you take out the second term, that's the Hopfield rule. Or the basic associative memory rule.
Cool.
And so this extended form also existed before our study, but what we did, well, this extended form, except for this sort of thing, this idea of putting it in a memory graph, and they were just doing like plus one onto mu. So they were putting, they were making like this one was mu plus one,
I see.
or something like that.
Mm-hmm.
And so in our case, we're putting this into this memory graph, and then we're formulating the equations. And the other part that's new for us is these little guys, and also the fact that we had these as well.
What was
so
C?
I suppose actually C is just a parameter in 0 to 1. And so you set that at the beginning of your simulation. You could modulate it over time, and we think in the brain that you probably would be modulating it, but it sort of controls how much, like a balance between the global and the local inhibitory activity.
Yeah,
so this is close to 0. Sorry.
Yeah, I'm interested in how inhibition comes into these equations, because I was going to ask you earlier how I think about inhibition in the context of Hopfield networks. I guess it naturally you'd think of modifying the connection weights or something. But
so in this case we have explicit excitatory and inhibitory weights, so they're entirely separate. Whereas in the traditional Hopfield network, they're sort of blended together, aren't they?
Yeah.
So in this case, we have like this this green part is entirely the excitatory activity going back to itself.
Yeah.
So this is kind of the excitatory to excitatory part. This is like, and these parts are inhibitory towards the excitatory, and then these dynamics down here are just like showing the excitatory to an inhibitory parts. And it's important to note that there is also an inhibitory to inhibitory type of connection, which we don't model.
Sorry, so the superscripts E is for excitatory, G and L stand for different kinds of inhibition. Is that right?
Yeah, exactly. G is global and L is local.
Oh, right. Yeah. Mm.
Yeah. Yeah.
Interesting. So there's like sorry to interrupt you. In LSTMs, there's like various kinds of gating that modifies activity based on, you could say, excitatory activity, I guess. I'd never thought of in like transformers decomposing attention or something in a way that models both excitatory and inhibitory. Is there like these two things, they really are essentially different? I mean, there's a sign difference, of course, and I guess in your model the C's can only be 1 or 0, so they can never take negative values. But I mean, once you have a differential equation, maybe it just all gets mushed together and it's hard to tell that there's really a difference between excitatory and inhibitory, but you really see that they behave quite differently, these.
Yeah, well, in the brain, these neurons are totally different, and their behaviors and implications for disease and all sorts of things are different. And so on that basis, we think that they probably have a special computational role that are different than the excitatory neurons. And in order to tease that apart, we first have to sort of disentangle the excitatory from inhibitory connections, which is part of what this model does. And then the other part is, well, let's see what happens if we just have at least 2 different types of inhibitory connections. And in this case, they're sort of just functionally connected to the excitatory neurons in different ways, otherwise being dynamically, similar. And then we see the effects by modulating the balance between them with the value of C, which can vary between 0 and 1. So you can have like 0.5 that would be a balance between both. And in terms of like the implications for, let's say, a transformer or something like that, well, we're not entirely sure what implication that will have for like these results might have for that sort of system, because of course they're all jumbled up if you follow down that track. But it's unsure basically. It could be the case that it would be better to make transformers with explicit inhibitory and excitatory neurons.
Could I ask? I mean, you could have formulated the many different models in order to incorporate inhibition. Here you're having a different sum end with a different sign, and that is natural. You could have chosen to do other things. There could have been multiplicative factors, for example, in the formula for TIJ. Yeah. What motivates this particular choice? I mean, it fits the data better than other possibilities, or it's just one of various things that people have tried? I
suppose it was like the simplest. I thought it was a fairly simple way to go about things initially, at least. There is evidence that these things have a multiple, like in the biophysics, there is a indication that some of these interactions are multiplicative, but in general they're more additives or subtractive. So they're like, you get a quantum of excitation and inhibition, and they sort of cancel each other out. Whereas there are other cases where there's like a neuromodulation that happens, and that can have a nonlinear effect. And in that case, it might make more sense to have like some sort of more complicated system.
Okay, but it sounds like there's some good reason to think that it's additive.
Yeah, I think so. Yeah.
Cool.
Yeah. And so on the right side, this is just like an example simulation. The y-axis is time. The y-axis is the spiking rate, and the x-axis is time. And you don't have to worry about the units for the spiking rates. You can just treat those as arbitrary units for now. And we've highlighted different groups of cells which are active. So the green one is showing the assembly for the stimulated pattern that we start the simulation with, and you can see that that's sort of maximally activated for about 80 milliseconds or so, and then we turn off that stimulation and let the simulation sort of just run to the end. And you can see it sort of is stable at this point with green, with the green activity being highest, and that dark line is the mean, and the shaded region is the standard deviation of all the neurons in that particular assembly. And then in this case, we're actually looking at a memory system which is like a circle. So we've stimulated one here, and then we're looking at the first neighbor and the second neighbor and the third neighbor, and we can see their activities here, which are getting lower. And I've forgotten what value of C this is, actually, but this will be for some particular value of C.
So what happens if there's no inhibition? I mean, if it's a Hopfield network, you'd recall, like, I mean, why wouldn't something similar happen if it was just not involving inhibition at all?
Well, if there was no inhibition, all of these particular things wouldn't have a reason to ever go down. So, like, this one would just go up and up and up and up. This one would just go up and up and up. These would all just go up.
I see.
And so you would have sort of like delay of these things going up without any control.
So if I ask how to fit this into my understanding of how a Hopfield network recalls a memory, should I think of this as being so you stimulate a pattern, and that's like passing some activation through that second summand in your TIJ formula to nearby patterns, and this would just keep recalling everything unless I had the inhibition. So it's like the inhibition is doing something to cancel out the effect of that second term, or like how do I modify my understanding of recall in a Hopfield network in this modified system?
Yeah, sure. So I like to think of it a bit like, because we never turn off because the excitation basically never dies in this model, I sort of think of it as fire, and like different fire departments dealing with this fire, like locally or like calling on God for it to rain everywhere. And then, so when we start the fire in one particular memory pattern, which is like fire is like excitation, you're starting it there, and then it's spreading to its neighbors according to the memory system. And if there's enough sort of like local resources in the inhibition, like it's balanced that way, if C is sufficiently close to 1, then you will be sort of dealing with that fire locally, and it won't spread that much. And but what will happen is that the total amount of fire can get very high, because you don't have this global inhibition that's happening, because you've also got random activity, like random sort of sparks of flame like everywhere going, not directly from the fire but just from the noise. So this actually this should have, there it is, right? This is the excitation. So actually, yeah, this shouldn't be colored purple, this part. Maybe just let me put a white thing around that. So this part is the excitatory, is where we put in the stimulation, but we use the same term for random noise.
Right.
And since there's random noise going everywhere, there's kind of like sparks of positional fire that come up in other places. And if you don't have sufficient sort of rain or global inhibition, then those fires can sort of spread into wildfires, and you can lose control of the network.
Cool, thanks. That's a helpful analogy.
Okay. I apologize. It's almost been an hour. I'm happy to keep going. We've got a few more slides, but feel free to stop me at any point.
No, okay. Please finish. Yeah.
Okay. So let's go on to the next. Okay, so finally some of my results. On the left side here is that same memory system of a circle, but now we're varying the value of C. And in panel B, you can see different values of C starting at 0. So that's 0 would equate to global only. And then as we're increasing C, we're getting more and more local inhibition involved. And this black line is basically, so each of these gray lines is kind of like what you saw from that paper in the monkey with the fractals. And on the x-axis is the distance or the number of hops away in this structure here. And you can see that basically this line and so the grey lines are different trials, the black line is the average of all the trials. I think there's 100 trials.
I'm a bit confused about the 4 different graphs. What does 4x mean?
In C?
Yeah, that's the one you were talking about, wasn't it?
Sorry, I was talking about B.
Oh, you were still in B. Okay, sorry. So I misunderstood.
Yeah. It's okay. But yeah, in B, the message is just that as you increase C, as you increase local inhibition, then you get a movement to the right in terms of this this line. And
so to go back to your fireman analogy, so if C is 0, so that's purely global, you said?
Yeah, purely global.
So then that's just raining all the time, so no fire.
Yeah.
Yeah.
Right. But then if you sort of pull back a little bit with the rain, but then let the local fire departments come out with their buckets or hoses or whatever, they don't do as great a job, but they do control it somewhat, and then it spreads, but not quite as far as it could. And what's interesting is if you put this up to 0.7. So this is getting close to C equals 1, which would be completely local. You have a problem, which is that you have like this crazy activity going on. And this 1 times means that the original network is the same size as it was for all these other ones. But then as we increase the size of this network twice again, and finally twice more, we get a cleaning up of this activity. So we think this is evidence of a finite field effect.
What does increase the size of the network mean? Just the number of neurons?
Just the number of neurons. Nothing else changed. Yeah. And so we didn't formally test, why this occurred, but we just, based on this, thought it's probably a finite field effect. But what's quite nice about this is if you compare it to the original thing, if we sort of put a measurement on like what's the number of neighbors away that this thing goes basically to 0, then plot that in this sort of picture in D, then you get, as you increase C, you get a correlation distance which goes up to approximately 20, which is a lot more than 5. And of course, this kind of, this might not be always useful, right? You might want very specific recall of particular memory patterns, not like broad association in time or space to things 20 hops away from you. But when you do want that, or maybe, for example, when you drink alcohol or things like that, your inhibition sort of like gets a little bit more out there, and you're making these wild associations, and can have creative moments or something like that. Or maybe just creative people have an ability to control this sort of inhibition, or the natural inclination towards local firefighters compared to praying to God for rain.
Yeah, that's cool. Are there any like sudden transitions as you vary C? It looks pretty gradual, but you could imagine in systems like this there would be a phase transition at some particular value or something.
Yeah, it seems pretty gradual, except for the fact that suddenly it can go berserk in for example, C equals 0.7 if the network is not large enough.
Mm-hmm.
That seems to be the only thing of my playing around with this that sort of has a phase transition, and that could indicate that like this is a phenomenon that only works within some ranges, and outside of that it sort of doesn't work, or maybe it needs to be modulated by something else, or
Mm-hmm.
I don't know. And so the next slide here shows some more interesting memory graphs, and here the arrow points to the memory that we activate at the beginning of the simulation, and how dark these different vertices are represents how active those particular assemblies are. And you can see that in general the activity spreads. You can think somewhat geometrically from in terms of the graph distance from that first vertex away, but there is also some topological information which is kind of getting picked up in, for example, this example here. You'll notice that we in the bottom row you'll see that we activate this particular sort of assembly or memory item, which is neighboring to a room on the right and is within the room on the left, and yet the activity sort of consolidates within this room rather than, say, spreading out into this room a little bit. Also too, this is the TUT graph on the third row, and we simulate the middle. And in this, and sorry, I should just also mention or highlight that this is with two different values of C. So this one is more in the middle between, the one on the right is more between, it's slightly more towards local, and then the one on the left is more global. And with the more local ones, you see generally a larger spread, but you also see some interesting effects, and so not just about geometry, it seems, in the graph and what I call the four rooms graph at the bottom. And then we can also sort of do a correlation matrix between the activities of all these different neurons, and order those so that we can sort of just visually inspect whether or not these are sort of clustering together in any way. So you see, for example, on the right side in panel B, that we have two columns again for different values of C, and these are basically the vertices ordered in some particular order.
The blocks are the pentagons, I guess, but where are these two black dots that are escaping to the other pentagons?
Sorry, which black dots?
Looking at the top diagram in B with the three black blocks, I assume those correspond to the three pentagons.
Yes, they do. Yes.
So what activity correlation? I see. So this.
So
yeah. Okay.
It's at the end of the simulation. We can given different starting points, we can look at and given all the different starting points, we take the sort of normed activity correlation over all those, and then we can come up with a picture like this. And in that example, these like weird things which aren't exactly part of the squares are the sort of edges of these things here. Yeah. yeah, you can look into some of these, as I have for a long time, and I only tested these 4 different graphs and the circle. So you could do a whole bunch of other tests with different graphs, which I haven't done.
So can I probe your intuitions here a bit? So you're looking for a sort of model with this inhibition coefficient and tuning it appropriately. You kind of want to see what's in this right-hand column, do you? Where you sort of think that the neurons are arranged in some sort of topological / geometric / graph theoretic way that reflects something about the world, and then you kind of recall all the bits in a component or something? Is that what you want to see happen somehow?
Yeah, something like that. So maybe just to say a bit more about my intuition. So like the 4 rooms, for example, I analogized like 4 rooms in some navigation task, and these different memory items could be place cells. So people here might know what place cells are. They're sort of they're areas in which a particular cell in hippocampus will become active at that place. And in this case, in the TUC graph, you could think of these as 3 different rooms, these clusters here, and then there's like a central doorway which leads to these 3 rooms, and you're to make a choice about which one to go to. And in the case of global inhibition, we're quite quickly making a choice for one of the rooms, but in the more local inhibition case, we're quite quickly not making a choice, and we're sort of keeping our options open for going into any of them. And I'll show more of the dynamics of how this particular one evolves over time in the next slide. And so in general, these things on the right, the correlations between the different activities of neurons is a bit like, sort of just summarizing how these different memory items might be related, and how those relations are sort of being modulated or emphasized by the inhibitory activity, even though we haven't touched the excitatory. So I think what's cool about this is that we're not changing the excitatory activity or connectivity whatsoever. We're just modulating it indirectly through the inhibitory balance, and we're not even changing the total inhibitory strength.
So you could, maybe I should save my questions for later, not to take up all your time. But I have a question about the relation between inhibition and just temperature. I mean, you can of course dial up the transitions just by raising some kind of thermodynamic temperature and just transmit more excitations all the time or less. But inhibition is sort of like making that directional in some way, or I mean, how do you think about the relation between just temperature and inhibition?
Yeah, it clearly is related, and in a very coarse-grained way, it is like going back to sort of the yin and yang analogy, or blanket of inhibition analogy for inhibition. However, how that sort of gets operationalized in the specifics of like a particular type of inhibitory neurons, which are globally or locally connected, let's say, I think matters. And like the temperature could be different in different areas. So like you've got maybe that's kind of what's happening if we have a purely global inhibitory dominated state, then we've got pretty constant temperature everywhere. Whereas when you have local inhibitory activity, you've got non-local fluctuations. Non-global. Yeah, you've got local fluctuations, and then maybe those can get out of control, like we see if in panel C on the left. Thought provoking. So shall we move on?
Yeah, that's good.
Yeah. Sorry that I probably put a little bit too much intro.
It's great. And also, I've been asking lots of questions, so I don't think it's like
No.
prepared too much stuff.
Okay. No, it's really nice. I appreciate it. So we're nearly at the end. On the left is the tut graph over time. So here I've sort of binned the activity into little sections of the simulation. These are milliseconds, like little 20 millisecond groups of the simulation, just the average activity. And what's nice to me is that in both cases we stimulated the center one first, and then we let the activity spread. And in the global one, you sort of see that the activity is slightly higher actually on this particular sort of cluster, and it quickly sort of has like a winner takes all scenario where this one just takes all the activity. Whereas when you have so this is the more globally dominated one, and this is the slightly more locally dominated one. And in this case, you sort of have an initial spread of activity, which is, first of all, larger. You can see that these activities are, for example, fewer than on this side. So the geometric spread is larger, but also it's sort of maintained throughout. There's no real change. There's sort of like a winner shares all scenario. And in panels B and C, these correspond to the global activity and the local inhibitory activity of all the local and global groups. And the global group is just one. Each grey line here is a different trial. And these are from one trial, and these are all different neurons. And you can see that so this one here is for global, the global one. This one here is for local. And you can see that in the global case, there's these kind of three modes or steps that are happening. And those kind of correspond, it seems, to the fact that initially we have basically a different number of neurons which are active altogether. So at the beginning, perhaps there are fewer, and then there are more and more, and eventually it sort of saturates. Whereas in the local case, it sort of goes all up together and stays flat. And then these different local groups or local units of activity are all sort of like complicated in the global case, because you've got these ones which eventually losing out.
I'm not following these graphs. So you've got global and local in B and C respectively, and then within those you've got global and local. So what is...
sorry. So this one I mean C equals 0.1 and C equals 0. Sorry, that was confusing. 525. So I just mean in terms of the value of C in the top panel. So in B, these panels are relating to maybe I do this in colors. So this one, and then this one.
So in B, what does global mean? Like, what's the first graph?
So that's like in the model we have global neurons and global inhibitory neurons. Those are connected to all the excitatory neurons, and the local inhibitory neurons, which are connected only to specific memory items or memory assemblies. And we're tracking the activity of those neurons over time.
This is a graph of the values of these inhibitory things, like how much inhibition they're doing. Yeah, okay.
Exactly. And so the reason why this, in the case of the green sort of case, the reason why that has this sort of step-like structure is because over time the number of sort of fires that are happening, or sort of the intensity of the fires are sort of changing over time. And then also the local fire departments are like coming and going because there's different things going on. Oh, okay, this is the first time to use the razor. And in the local case, everything sort of starts in a particular way and ends in the same way. it seems.
Interesting.
And then finally, I regret not putting the equations here for how I calculated these indices, but you can look at the thesis for how they're calculated. But basically, the higher these different indices are to 1, the more that they correspond to a particular other type of labeling, which are just sort of where you label this memory graph with 1s on some subset of the graph and 0 everywhere else. And in the case of the clustering indices, we label them according to like whether or not they belong to different clusters, and we pick the one that's best. So in this case, for the clustering indices for the 2 different values of C, this, by the way, this diameter thing is just the diameter of the graph, just graph statistic. So the clustering indices are based on this thing called the label propagation algorithm. And in this case, basically the final activity of the network after simulating with C equals 0.7 for this particular graph, and its overlap with the labelings that you would get if you used the label propagation algorithm is 0.6 approximately. So that means that for this particular graph, if you use this label propagation algorithm, which you would suspect sort of cuts them into clusters like this, then you would see that there's a general overlap with the activity in that way. That the correlation would be 0.6, and the reason why it's not quite as, let's say, 1 is probably because of these little guys here, these activities which spread across into the other clusters.
So sorry, maybe you said this and I was thinking, but so the label propagation algorithm is just some known graph clustering algorithm, and you're sort of making the suggestion that this kind of local update rule, Hopfield style, is a sort of way of computing that locally. It's something similar to label propagation or something like that. Is that?
Yeah, it could be. And actually, you'll notice that, for example, in this first case, so let me just label this case number 1 and this case number 4, and this is 2 and 3. In case number 1, in that graph, you've got the case that the more globally, like the C, when C is lower, and you've got more global inhibition, the overlap or correspondence between, this rule and the dynamics that occur is actually more corresponding with the label propagation algorithm. So this value is higher than this value. And maybe since a few of us are mathematicians here, I'll just do this value and this sign, but this actually swaps here. So it's not consistent necessarily with this absolute value of C, but rather it seems to depend on the structure of the graph. And possibly also the choice of where you start the simulation. So if you chose to start simulating in some other, vertex, then this might change. And this one is approximately equal, and this one is larger. The geometric indices are to do with graph distance, and what we did is we looked over, so we were starting at some particular vertex and then measuring the distance out from that, and then basically correlating with the same sort of situation as the label propagation algorithm, but then for different distances, which are in brackets here. D equals 1 means like a distance of 1 away, and labeling all those as 1, including the original vertex, and then everything else is 0. How much does that correlate with the activities seen after a global inhibition simulation? And here for D equals 2 in the local case. And the reason why we've used different Ds here is we're just showing the maximal overlap one, so the one that's closest. In general, you see that for all of these, it's the case that the local inhibitory situation is having the highest value, even though the values, and also the values of D are strictly increasing, or non-decreasing. Yeah, this one is 3 and 3, but at least this value increases. So it seems that these dynamics and this rule is, occasionally picking up something to do with topology, structure of the graphs, but very much is increasing the sort of geometric spread of activity.
Can I probe a little bit? I mean, this is I could believe all sorts of connections between some update rule like that and various properties of graphs, but I suppose I have no idea how to judge whether or not this model is actually a good model of what happens in real neurons. How do you test that exactly? That seems like it must be quite difficult in general, right?
Yeah, well, we make some guesses as to which types of neurons these local and global inhibitory groups must belong to, or must have some properties of, so like biological properties, for example, like what they look like or how you could detect them. And so we've actually suggested, or I've personally suggested, some different experiments to experimental groups that would be able to do experiments, for example, like knocking out, that is like silencing or removing these different groups, or tracking their activities independently. And then on that basis, you could perhaps see that these different inhibitory groups are behaving like the way that we describe, or they might be behaving differently. And, you could then do a whole bunch of behavioral tests. Probably you would start with something simple, a bit like with the monkeys and the fractals. But yeah, you could do, I think, much more sophisticated things to do with, associating memory items which are very far in the past or future, and tasks which might rely on that, as opposed to maybe other tasks where you have to be very focused. And so you might also find that different drugs affect this balance quite a lot in the activity of different inhibitory neurons, and then that would also create a benefit or a detriment in different tasks. And then as for, let's say, transformers and attention, I think, although I haven't made the rigorous connection yet, I think it's possible that you could use some of these ideas to help with, like, connections of different scales in, let's say, language, the idea of a paragraph or a page or a sentence or a word. These are sort of different scales of things that just do with the distance between the words, but also, I don't know if there's like a topology of language, but yeah, I suppose that vaguely there could be.
Yeah, now we'll have to sit and talk for an hour about that. But so where did the scale come from? That's the patterns, I guess, as you're thinking about individual
the scale
neurons.
Thing, yeah, the scale,
patterns,
yeah. So the scale would just be like. Do you mean the scale in terms of like language that I was talking about?
I guess I can sort of get where you're going there, but you just introduced this idea of scales, but you're sort of building that out of what you've just been talking about, I guess, in some way. Yeah. So I don't understand that connection exactly.
Just kind of like, well, for example, if let's say in this on the left side of the OrbCam, this slide here with this 4-room graph, I could. I have 4 rooms in this picture, but I could like break those up into 4 rooms further, and again and again recursively. So, I assume, I didn't test, but I assume that this algorithm or these dynamics would have some funky, interesting effects when you break up the memory system or memory graph into different scales, and maybe these effects would, you could maybe modulate or stack these effects on different scales as well.
Mm-hmm. Yeah, very interesting. I mean, this kind of thing is in the direction of block spin renormalization and some of these ideas that I suggested I was interested in last time we talked. So we should definitely talk more about that, I think.
Yeah. There's actually also, just on this very particular point, there's also a great paper called Hierarchical. I'm not going to be able to spell this, probably. Hierarchical. I'm horrible at spelling. Hotfield.
Oh, really? Okay, that's interesting.
Networks, yeah. And that's that's in a journal called Neural Computation. Oops. That was released a couple of years ago, and that's an interesting paper, kind of coming into that idea of multiple scales of things, and multiple scales of memories. Yeah. Interesting.
But I will let you get to your final slide.
Yeah. So I think, just in summary, then there's local and global inhibition here in this model, and that sort of seems to affect the extent of the recall in these sparse assemblies. I suppose I didn't emphasize that, but they're also quite sparse, whereas previous models are not sparse. That is the activity of these neurons, and these activities are sparse, which is more realistic. And it seems to shift the topological or geometric focus of the activity in these different structures that we tested. And that's the end of the presentation. Thanks very much for your attention.
Yeah, thanks a lot, Tom. I will save my questions because I can ask next time. But Ben, Chad, do you want to ask anything?
I unfortunately disconnected for 20 minutes in the talk because my computer crashed.
Oh, no, okay.
Yeah, this was actually during when you introduced the model over here.
Yeah.
I can probably go back and watch the video, but maybe just for me, just quickly summarize what this TIJ is supposed to be. So,
TIJ is the connection strength between excitatory neurons. So in this model, we have the excitatory neurons, which their total number is n superscript e, and then
sorry, so I can see this as being the connection to the using model that I was promised.
Sorry, could you say that one more time?
I assume this is like a similar idea to the async model. Like, are these?
Yeah, exactly. Yeah, so we have so this is exactly like the async model, and this first part, let me denote this first part like A, and the second part B. So the first part B, A is having like infinite range connections between anything which are within a particular pattern mu, and then it doesn't matter how far away they are. But then with B, we say, okay, well, we're going to add some extra interaction strength if you share activity with neighboring patterns, and those patterns I'm talking about are in some memory graph. This is like one such memory graph, and K is the set of vertices which are neighboring some memory pattern mu. No worries.
Let me just check the chat. Okay. Maybe there's no more questions. So what's next time, Tom?
Next time, I think we're looking at simplicial Hotfield networks.
Goody. All right. I guess we'll leave it at that. Yeah, thanks a lot, Tom. I'm looking forward to next time.
Yeah, thanks very much, and apologies for being a bit over time.